Hi all, welcome back to Microbiology Lectures, and today we're going to talk again about microbial growth. Last time I introduced kind of a growth equation. It's a very it's an equation that's kind of universal. Anytime we're talking about exponential growth, that's the equation that we use, whether we're talking about bacteria populations of ants or fleas or trees or whatever. It doesn't really matter. And so we'll talk more about what goes into growth, population growth, and things like that. Before we get into that, though, I want to make sure that it's kind of clear that microbial growth, just like if we were raising animals or plants or fungi or protozoans in a laboratory, it's very different in nature, right? And so the growth that you see in nature is different for all organisms, but microorganisms is especially we grow them under conditions that are suitable to us. So a uniform temperature, uniform nutrient type, uniform humidity, things like that. And they are very different. More so than a lot of other organisms, their morphology is very different from the laboratory to nature. They have very different actions and they produce different products and all kinds of things. So a couple things is when you change conditions, and you can change them in laboratory also, but it's more difficult to replicate natural systems in a laboratory because we'd have to control for light, temperature, humidity, wind speed, the moisture level. If they're in aquatic systems, the flow of the water. I mean, there's all kinds of things that you would have to control for to try to get a natural population to grow in a laboratory. It's very difficult. So normally in laboratory conditions, we don't do that. So when we look at their behavior or their growth or whatever, it's very different in nature. With most bacteria that we study forming biofilms or being associated with other bacteria in nature. So a lot of times these bacteria that we're studying in a laboratory, when they're in a natural system, they might be associated with a lot of different bacteria. They might make a slime coat, or their capsule might be a different structure, and they might occur in biofilms, and they might exchange chemicals with other species and all kinds of things. So just knowing that our growth curves that we often look at, like the one that I showed you last time, where it's like, okay, here are the hours or however long for it to go from the lag phase to the exponential phase to the stationary phase. Just having that information from a laboratory organism is very different from having that information for a natural system or an organism that's living in a natural environment. Okay, so they can adjust based on their surrounding. They produce materials that are appropriate for the situation. Okay, so if they need to produce toxins to prevent being consumed, if they need to produce intermediate molecules and pass those to neighboring bacteria to help with metabolic processes, they will. And we just don't often see that cooperation in laboratory conditions that we see in nature or natural conditions. So again, most of them are going to attach to a surface or something, whether that be inside an organism, outside, like on a rock, or in human-made processes like on concrete walls or sinks or pipes or just all over the place. Gutters of your house. I mean, there's bacteria colonies everywhere. Often they'll be in a community of other bacteria that can live in the same environment, and they produce biofilms. And so, just some of the things that you might be somewhat familiar with from a biofilm situation is a lot of the slipperiness on rocks or even concrete that's under wet conditions that often have maybe leaves or some kind of organic material that might provide food source for the bacteria for that biofilm. Or maybe it's in a stream bed, like along a lake or something like that. That slipperiness is typically the slime layer from different species of bacteria that are forming a biofilm. Okay, I talked about this before. We often think, associate bacteria with humans because humans often produce enough energy as waste products that lots of things can grow in our inhabitants, our homes, our buildings, and things like that, just because we have so much excess energy that we're depositing, whether that be in the drains from washing utensils and food and our hair and just all kinds of things, to waste products from our body going into toilet bowls and things like that. The plaque that occurs on our teeth, excess food and stuff like that they're consuming. All these things are biofilms, and I wouldn't say we're just beginning to know about biofilms, but every single kind of probably day or week we learn more about biofilms. We know about the association. We know, more often than not, the biofilm has multiple species in that biofilm, which means that they're cooperating with each other. In some cases they might be competing with each other, but often they're cooperating with each other to generate that biofilm. Maybe one organism is very good under rich oxygen layers. So if we take this biofilm here, we might say, okay, if this is on the surface of the water, maybe the organisms on the surface here of the biofilm are photosynthetic. Okay, but the organisms underneath it don't have to be photosynthetic. Maybe they produce a toxin, and maybe they get their energy from the photosynthetic organisms, but in return the photosynthetic organisms get some exotoxin that makes this entire mat of biofilm toxic. Okay, and so this is that cooperation piece, and we're beginning to start to study these cooperations and understand what's going on in these different environments. So biofilms, more about them. Again, they can have biological implications. They can be really, really important. Dentists will talk about the biofilm in your mouth constantly. The plaque that's leading to tooth decay and gum disease is, definitely related to biofilms of bacteria. But other than that, when we start diving in and we start looking at infections that happen in a medical sense, almost all of the infections are the result of some kind of biofilm. Multiple species interacting with each other that form on either a wound or in a region of the body that might have decaying cells or something like that allows for these biofilms to form. And then that biofilm itself can create an outer layer which makes it more resistant to antibiotics and more resistant to the immune system. And having multiple species next to you means that our immune system, let's say you have an infection, an internal or external infection, and it's a biofilm and you got multiple species of bacteria, it's harder for our immune system to target single species when the biofilm itself is multiple species. So it makes them more resistant to both our immune system and antibiotics. And so, of course, there's other concerns like accumulation in pipes, and a lot of those organisms that are accumulating in pipes and drains and things like that, they can produce toxins too. So you can get toxins that are popping up in the water. They can also degrade the pipe. They can degrade the seals of the pipe. So there's lots of things that are going on there. And it's not just pipes and drains and things like that. There's all kinds of things. Sidewalks and gutters and roofs and like all kinds of industrial concerns around biofilms that happen on docks and all kinds of things. The other thing is we get large biofilms that form on regions where maybe there's large amounts of nutrients being deposited, and then humans want to utilize these areas and not get sick. The more biofilm there is, often the more resistant they are to our disinfectants and our cleaning methods. Okay. But in general, they can also be helpful. So there are lots of ways at which we use biofilms constantly to benefit us. Okay, our wastewater treatments, almost all wastewater treatments have a biofilm application. So they have these holding ponds, and the biofilms form on these holding ponds, and they consume the excess nutrients and bind it up, and then we can clean the water enough to be able to dump it back into a river system without having huge amounts of nutrients. Enhance plant growth and soil nutrients, and then gas and oil production plus cleanup. There's just a whole range of biofilm interests from lots of different industries. And so some are preventative, like healthcare and dentistry, and then some are utilizing those biofilms, like agriculture, waste management, that kind of stuff. So just keep that in mind when we're progressing, that not all biofilms are a bad thing. They're natural, and in fact, most bacteria that we see in natural systems in their native habitat are in biofilms, and so it's their native way, and we don't replicate it very often in the lab because we're not sticking multiple species together that form a biofilm together, and they do so in nature, in the natural system. Alright, so just to hit on that again, so often these mixed microbial communities in these biomes, their interaction is cooperative often. Okay, so under the biofilm, they're cooperative. Okay, and that might be that they share nutrients. They may be sharing things that one organism can't process and the other one can. So like we know things like, so E. coli in our intestinal tract, E. coli can be grown and grows very well under oxygen conditions, so in the presence of oxygen. But almost all the other bacteria that you have in your intestinal tract that are really beneficial to breaking down different chemical bonds and helping you break up nutrients and deposit the nutrients, they're anaerobic. They don't do very well in oxygen situations. So even though E. coli may not be the main class of bacteria that is helping you digest your food, in essence, they're consuming the oxygen in your intestines, so the other bacteria can survive. And so that's a piece of that cooperative process, that you have these close associations between different species of bacteria, which allows for certain individuals to exist. And so again, you can have a situation where one's consuming oxygen, a situation where maybe one's breaking down an iron or breaking down sulfur or something like that, and then the byproducts of that is passed to another species, which then can utilize what one species might think of as a waste product, the other one might think of as an intermediate to a product that they might need for an energy source, or maybe to develop a toxin or something like that. Okay, so the interaction is often cooperative, but it can also be competitive, and there's lots of ways in which they can compete with each other. And so they can compete by producing antibiotics. So often in lab, that's what we're looking for. Like, we're trying to find bacteria or native organisms that might be in soil or something like that can produce an antibiotic. And then does that antibiotic prevent growth of Staphylococcus? Does that antibiotic produce or reduce growth of other bacteria that might be of interest to us? Okay, and they can do that by a couple ways. They can synthesize toxic compounds to inhibit competitors, and sometimes that is just passed by basically driving a structure into the other organism and depositing it in them. So it's like you're killing the organism, or sometimes it's just expelling these exotoxins around the outside of the organism as it grows, which doesn't allow things to come in contact. And so we talked about chemotaxis, and so these chemicals would be expelled, and other bacteria might sense those chemicals, and those chemicals aren't favorable to them, so they move away, allowing for the one organism to not have to compete for nutrients with others. And so you can also have growth inhibition, so they can like, maybe it's not toxic, but maybe they can take the nutrients or bind up the nutrients that another organism needs, and so that organism maybe is inhibited, their growth is inhibited, maybe the colony sizes are smaller in the presence of another individual. There's all kinds of ways at which you can get competition between two different species. So it works both ways. Lots of the interactions are cooperative in biofilms, and then there's also lots of interactions that are competitive, because anytime you have a situation where you have multiple species in the same environment, energy is limited, and so the nutrients available for that organism to grow is limited. And the more species you have that are trying to absorb the same nutrients or the same energy source, the more competition you're going to have. And we'll talk about this from a graph of growth. And so if you increase competition, then you should expect the growth rate of the colony to be affected by that. If you decrease competition, then you should expect the growth rate to maybe increase. Same thing, it's a little harder with cooperation to see these changes, but you would expect the same. Like, if you increase cooperation and organisms are able to survive and grow and increase the colony size with different species, then you would expect that growth rate to increase. If you decrease the cooperation and you remove an individual that might be needed for that biofilm to really thrive, then the growth rate's probably less. Okay, so when we come back, we'll talk about these growth curves in more detail.